in. Yeah, so this is the weekly overview page for week 1. So again, those weekly overview pages are always a good place to go to start your week and to see how to prepare for the next week. And so you can see the things that are due there. So that lecture quiz 1 is due on Friday night by 11:59 p.m. If you click on it, it'll tell you what it's over. Okay, so they're always over 2 sets of notes. So this one is going to be over what we covered yesterday in class and today. Okay, so visual system 1, and then the sensory receptors and spinal reflexes that we did on Monday. Okay, so that'll be what you want to study, those 2 packets to prepare for the quiz that you'll do on your own before Friday night. Okay, so after today's class, you have everything you need to take that quiz, so you can take it whenever you're ready. All right, and then if we go to the week 2 overview page, I do just want to show you this. We'll have an IRAT and TRAT on. That'll be our first day meeting in person, so that'll be a fun way to start it off on a TBL day. So you guys will sit with your teammates. I'll send you some information ahead of time so you know where to sit. But in order to prepare for that class on Wednesday, you'll want to watch the video that I will have posted. So right here for Wednesday's class, you'll see a link to a video. I'll put that up after this class. That'll be just like me going through the notes, just like I would in class. You want to fill that out, study that very well, come ready to take a quiz on that when you come to class on Wednesday. And you'll turn in those quizzes via the clickers, okay? So make sure you have your clicker with you on Wednesday. Okay, so that's, and like I said, I'll put that link to that video in there on just today after class so that you'll have that available to you, okay? All right, any questions on any of that? All right, and then again, next week will be our first lab too. So if we go back to the week 2 here, so reflexes, sensory receptors, and cranial nerves will be the lab. It says the lab quiz here is over the cranial nerves, okay? It says bring handout, and again, reminder of where to find that handout. It's over here and under the modules, you see all the lab handouts there, and they're in the right order. So that first lab handout, that reflex and CN lab, that's the one that you want to print up and have ready to bring with you on lab for lab next week on Thursday. All right. Okay, well, let's go ahead and hop into the material for the day then. Today we're getting into the special senses. And so we're going to start out with talking about you know what, I think I just realized something. Okay, here we go. All right, so we're going to get into the eye today. And so one of the most amazing sensory organs in the body by far, and just some kind of incredible facts about the eye. So out of all the sensory receptors in our whole body, 70% of them are found in these 2 little eyeballs that we have, right? So 126 million sensory receptors between our 2 eyes, okay? Somewhere around half of our cerebral cortex, which is the big part of our brain, is involved in vision in one way or another, right? So it's no exaggeration at all to say that humans are vision dominant creatures, okay? There's many other creatures that are chemical sense dominant, like they smell really well, they taste really well, like dogs, cats, and most other animals are chemical sense dominant, but humans are by far visual sense dominant for sure. Okay, so today is going to be primarily anatomy of the eye and looking at the different structures that we find in the eyeball and around the eye. And then Friday's class, we'll get into some of the physiology of how all those different structures work. So just some of the accessory structures first, so things we kind of find around the eye. So first of all, the eyebrows are important for providing some shade to the eyeballs, as well as catching sweat as it drips down our foreheads, okay? Our eyelids are controlled reflexively. We can control them voluntarily, but they're also controlled reflexively, kind of forcing us to blink every about 3 to 7 seconds. And the purpose of that is to spread lubricants across the eyeball. Okay, we'll get into to the glands and the lacrimal gland in a second, the lacrimal apparatus that spreads tears across our eye, but it gets some help in doing that by the eyelid. All right, and then we have what's called the tarsal glands, which are little exocrine glands, and you can see that here in this image, kind of up here in our eyebrow. They secrete this oily substance that helps lubricate the eyelid. Okay. And so one of the accessory structures of the eye is known as the conjunctiva. Okay, and these are different mucous membranes that line the inside of the eyelids, as well as the white portion of the eyeball. Okay, and the purpose of this is to release some mucus to keep the eyes moist. Okay, anytime we irritate the conjunctiva, that's what's known as conjunctivitis. Okay, just that itis always means inflammation. Okay, if this irritation is caused by a bacteria or virus, this is what we know of as pink eye, right? Like this guy's got here. Highly contagious, especially if it's the viral kind, and can be pretty, it can be pretty tough to deal with that viral kind. Can take a couple of weeks to go away, and there's not really a whole lot you can do about it if you have the viral one. If it's bacterial, you can just get those antibacterial drops, and they go away super quick. But the viral one, if you've ever had that, it can last a week or two, and it's just nasty. Yeah, it's just your eyeballs get crusty every morning, and it's super uncomfortable and not fun. But yeah, so that's conjunctivitis. Okay, so more accessory structures here. This idea, this thing called the lacrimal apparatus is multiple structures together. And so we can see it starts up, or it starts here actually in the lacrimal gland. Okay, so you can see that gland. That gland releases tears in this kind of superior lateral portion of your eye. And then you can see they kind of flow across this way with the help of your eyelids. And then they go in to these little ducts here. Okay, so there's like a little hole right here connecting your eyelid to your nose, right? And that's called the nasolacrimal duct, and that connects your eye to your nose. And so this is why when you cry, you get a runny nose, right? Because those extra tears are drained out through your nose, and that's just the normal path. And so normally when you just have a normal amount of release of tears, it's not an issue. You don't even notice that because you just kind of gets absorbed into your nose. But when you have excessive tear release, like when you're crying, then it flows down your nose a little bit. Okay, so there's multiple things in this mucus. Okay, in this in what it releases. So there's mucus, which is there for lubrication, protection, but there's also antibodies that help destroy microorganisms, and then there's also proteins called lysozymes, which are helpful in destroying bacteria that make their way into your eyeball as well. And so the release of these tears are stimulated by the facial nerve, cranial nerve 7. Okay, so this just kind of walks us through that conducting system that I just kind of did already with the pen up there. Okay, one thing that's interesting about emotional crying is that there's something else added to the tears that isn't normally there. And so they find increases in the levels of enkephalins in tears when there's emotional crying. And enkephalin is a feel-good neurotransmitter, right? So it actually, tears themselves actually can be absorbed into your bloodstream a little bit and actually cause some relief of stress, relief of negative emotion, and actually make you feel better. And that's part of the function of emotional crying. Okay, and then it's also pretty common, especially in babies, to have a blockage of this nasolacrimal duct. And so if you've ever been around a newborn, or if you work in a postnatal unit at some point, postpartum unit at some point, then a lot of times babies have these little really crusty eyes, and it's because they just have a bunch of mucus stuck in that nasolacrimal duct, and it's just not clear yet. And so that the tears just build up there and get kind of crusty. So you have to kind of massage the inside of their eye a little bit to kind of break that up, and eventually it clears up. But it can get infected if it doesn't clear up quick enough. Okay, so those are some of the accessory structures of the eye. Any questions on these so far?
I have a question.
Yeah, go ahead, Trevor.
So sometimes, like when people cry, their eyes get kind of puffy. Is that at all related to like inflammation of any of these like glands, or is that just like a skin reaction?
Yeah, it's a great question. I don't really know the exact physiology of that. I assume it's not necessarily inflammation, but maybe more related to just kind of a buildup of the tears, like in the conjunctiva of their eyes and behind their eyelids. But yeah, I'm not exactly sure. That would just be my guess. It's a good question. Yeah. All right. Okay. So yeah, let's take a sec. Actually, with this one, I'm going to let you guys take a second and do this. on your own. We're just going to keep moving on, but a lot of times throughout the lectures, I'll have these little kind of recaps, and so it's just an opportunity for you to kind of reflect on what we just talked about. And so yeah, I'll let you do this one on your own though, because there is quite a bit to cover in this set of notes today. So yeah, you'll just walk through and just kind of fill in with each of these accessory structures, where we find them and what their primary functions are. Okay, so you guys can go ahead and do that on your own. Okay, so we're going to dive into the external structures of the eye. And in the eyeball, we have these structures called tunics. Okay, and so tunics are really just kind of, we can also just call them layers, right? They're different layers of the eyeball. Okay, and so the most outer one is known as the fibrous tunic. Okay, it's very tough and gives the eyeball some structure. Okay, so there's two different portions to it. The white part of it is known as the sclera. Okay, it functions as the thing that gives the eyeball shape, as well as the solid attachment point for all of the muscles that are going to move the eyeballs. Okay, and then the front part of our fibrous tunic is the cornea, right? And so this is the anterior part covered by epithelium, both the internal and external surfaces. This contains these special proteins that are totally clear, right? So interesting type of proteins called crystalline proteins that are just completely clear. And that's kind of what's shown in these little images over here, the shape of those crystalline proteins. Okay, and so the function of this is really to begin the bending of the light as it comes into our eye. Okay, and then obviously for protection too, if anything's coming into our eye, we don't want it to be open to things just entering into the back of our eyeball. So this stops things from coming in. But it's also very important in starting to bend the light, getting it to focus. It doesn't really focus the light yet. The lens is going to be responsible for that, but the cornea helps to get that process going. Okay, if any of you have ever scratched your cornea, then you understand that there's lots and lots of nerve endings in that cornea because it's quite painful when you scratch it. When you scratch the sclera part, it's not as big of an issue. But if you do get a corneal abrasion, then it's extremely painful, and you're kind of constantly blinking and trying to close your eye, and yeah, it's not fun. Okay. All right, so that's our most external layer, and then we have this middle layer called the vascular tunic. Okay, so vascular means blood, and so this means there's lots of blood vessels in this region. Okay, one of the structures inside the vascular tunic is called the choroid. Okay, and the choroid is the posterior region of this, very blood vessel rich, and it also contains pigment cells, so cells that are producing proteins that absorb light. That's what pigment proteins do. And so the function of this choroid is to absorb light and to minimize light scattering, as well as supplying blood to the rest of the eye. Okay, one interesting thing that just to think about is that nocturnal animals don't actually have this choroid. They don't have this heavily pigmented layer. And so what happens is when a lot of light enters their eyeball, it just scatters like crazy. And it actually scatters in such a way that it just kind of, it activates all of the sensory receptors in their eyeball to the point where everything just looks white, right? So they're literally blinded by very bright lights. And that's exactly what happens with the whole deer in headlights thing, right? When a deer gets out in the road, looks up, and your car's coming, those bright lights go into their eyeballs. They literally become blind for a matter of 30 seconds to a minute, and they don't move, right? And that's kind of the idea, because they don't have that choroid layer in there like our eyes do. Okay, also part of the vascular tunic is what's called the ciliary body. This is the anterior part of the vascular tunic, and it contains smooth muscle, and this smooth muscle is under parasympathetic control, controlled by the oculomotor nerve, cranial nerve 3. And its function is, this is what contracts and relaxes to control the shape of the lens. And so this is what's going to help us focus light. Okay, and then coming off of the ciliary body, or kind of next to the ciliary body, are these little processes called the ciliary processes. And the function of these is they secrete this substance called aqueous humor. Okay, so the anterior portion of our eye is full of a liquidy substance called aqueous humor. And then behind the lens, or the posterior portion, is full of a jelly-like, more solid-like substance called vitreous humor, And we'll talk about that one in a second. But the ciliary process's job is to make and release this aqueous humor that controls the kind of pressure in the front of our eye. Okay, questions on any of that so far? Okay. All right, so a little bit about a little bit more about the other structures here found in the vascular tunic. We also have some other muscles besides the ciliary body. We have the iris here, right? And so the iris is the thing that gives our eyeball its color. Okay, well, those are actually muscles, and there's two different muscles within the iris. Okay, and those are both shown here. There's what's called the pupillary dilator muscle, which is this more external one, and then the pupillary sphincter muscle, which is this more internal one, which is a true sphincter muscle. It's circular muscle, whereas dilator muscle is longitudinal. It has longitudinal fibers that are just attaching to the sphincter muscle, and they can just pull that sphincter muscle open if it needs to. Okay, so we're going to talk about the function of those two different muscles here in a sec. But overall, the two muscles together, what they do is they determine the pupil size, right? By contracting more, by making the pupil smaller, we let in less light. By making the pupil larger, we let in more light. And we'll talk in more detail next class about why and when and kind of how that happens, but we'll talk a little bit about it today in terms of these two muscles. And so the pupil of our eye is really just a hole, right? It's what it is, and that hole is what lets the light in. Okay, and we need to be able to control how much light comes in depending on where we are, how far away the object is that we're looking at. Okay, and so normally when we want to dilate our pupil, okay, so if we start in the middle here, that's kind of mid-range, I guess. If we want to go towards dilation, we can see that pupil obviously gets a lot larger. Okay, there's two different ways that the eyeball can do that. Okay, one would be to contract this pupillary dilator muscle, but that one is completely under sympathetic control, meaning that we have to be really stressed out for that one to contract. Okay, so this is the part of the fear response. When somebody gets scared, you look at them and their eyes are kind of big and their pupils got really big. Okay, that's because when we're being chased by something that could potentially hurt us, we want to see everything we possibly can. Right? It doesn't really matter if it's that clear or not. We just need to see it. Right? So that pupillary dilator muscle pulls open our pupil really quickly when we're stressed out. Okay, but that's not the normal way that our pupil gets larger when we're relaxed and we're just trying to see something that's far away. Normally, all we do is relax this muscle in the middle. Okay, and then the opposite is true when there's something close to us. Our pupil needs to get smaller, and so it goes this direction into constriction. And so then what we do there is we contract that pupillary sphincter muscle. Okay, so this kind of walks us through it. So pupil constriction happens via parasympathetic activation using oculomotor nerve, cranial nerve 3. We can do that by contracting the pupillary sphincter muscle. Okay, when we contract a circular muscle, it gets smaller, right? It makes the hole smaller. When we relax it, it opens up. Okay, so we would do this for near vision. When something is close to us, we'll talk about this more next time, we need to constrict our pupil. I'll explain why next time. We need to constrict it when an object that we're trying to see is close to us. The further it goes away, the more we need to dilate our pupil. Okay, so we want to let less light in. Okay, and then normally, if that same object is moving away from us, now we need to inactivate cranial nerve 3, so then we can relax our pupillary sphincter muscle, and then that allows for the pupil to dilate, and then we can do better with far vision. We can let more light into our eye with something that's further away and there's not that much light from it actually hitting us. Okay, so this is kind of the normal way that it happens. But then again, we do have that second option in opening up our pupil really rapidly when we're under stress, and that's when we would use this pupillary dilator muscle. Okay, questions on that so far? Okay. All right, so we're going to do a series of questions here, and you guys can use the, I'm going to log off on my iPad just for a second and come back, but you guys can use your notes. I'm not going to send you to your breakout rooms because I that wasn't working. But go ahead. So there's this question here. And so really what we're doing here is just kind of a little recap of what we just talked about with those two muscles. And so the C and the R is kind of what you're choosing for this. So, right, so on that first group, we're talking about these two. This is the first ones, okay? These are the second ones. Okay, so for the first one, if you said that we contract the pupillary dilator muscle, then you would choose CD, that would be A. All right, so that would be A if you said contract and dilator. Okay, and then you go over to the second group for those two questions. Okay, so you can do that one. And then you can also label the structures here. And then I'm going to go over, to the next one. Yeah, and that's it for now. Okay, so go ahead and work on those for a second on your own, and I will get it so the poll will work for me. Okay, all right. Okay, so let's go ahead and put in your two answers just right next to each other in the chat here. Good. Okay, so we're got a few different things coming through, but most of them seem to be AD. That seems to be the majority. So let's take a look at those. Okay, so under situations of stress, we dilate our pupils via, okay, would it be contraction or relaxation? And then would it be the pupillary dilator or sphincter muscle? So under situations of stress, the sphincter, the dilator muscle is the one that's controlled by the sympathetic nervous system. So it's definitely going to be the D. Okay, so now we're down to either A or C. And then would it be contraction or relaxation? So that's a longitudinal muscle, it goes this way. So when we contract it, it pulls back, right? So A is definitely the correct answer there. Okay, but then the next part, but under situations where we need to see something far away, we dilate our pupils via. And now this one, when we contract a circular muscle, the sphincter muscle, it gets smaller. When we relax it, it opens up. Okay, so that's going to be the R there. Okay, so then it's either going to be C or D. And then which muscle? It's going to be the sphincter muscle. So that needs to be D. Good, so most of you put that. Most of you put A and D. Okay, any questions about that one? Okay, and we'll get into that more next week a little bit when we talk about the sympathetic nervous system and parasympathetic. And we'll talk about it more on Friday. We'll talk about the eye more. Okay, so this one, the tunic labeling, go ahead and here, I've also got a clicker question here. So which is the muscle that controls how much focusing is happening to the light that comes in? Go ahead and send in your answer on the chat for that one now. Good, so pretty much everybody is saying D there. Perfect, okay, so let's walk through those. So A is that front part of our fibrous tunic, which is known as the cornea. And then B is going to be that middle vascular part, the choroid. Right, C is going to be the retina. Talk about that tunic here in a second. D is the iris. Okay, remember that iris is made up of those two different muscles that we just talked about. And then E is the ciliary body. Okay, so that one's going to contract and relax to change the shape of our lens. Okay, and then F is the sclera, so that's that tough part of the eye. And then lastly, G is the optic nerve, which we haven't talked about yet, but that's where all of those neurons exit to go into the brain. Okay, any questions about any of that before we keep going? Okay, give me a thumbs up if you're ready to move on. Good, good, okay. All right, okay, so the last tunic here is what's known as the sensory tunic. This is the most inner layer of the eye, also known as the retina. Okay, there's two different layers to it. One is called the pigmented layer. This is the one that's a little bit further posterior. Okay, so it's the one that's up against the choroid. Okay, and so there's some pigment proteins here. Again, this also helps prevent light scatter, but it also is providing new pigments to the photoreceptors, because photoreceptors also have pigments in them, things that can absorb light. And so this is kind of the source of those new pigments when we need to replenish those in the photoreceptor cells. Okay, and then we have the nervous part of it. Okay, the part of it's actually nerve cells. All right, and there's three primary cell types within the retina. We have the photoreceptor cells themselves. These are the rods and the cones. Rods look at low light and black and white, whereas cones are more for high, more powerful light and color. Okay, these, when they get stimulated, are going to produce graded potentials. Right, we talked about these last semester. Graded potentials are just small fluctuations in the membrane potential. It could be in the depolarization direction or the repolarization direction. Not an action potential, right? Just a local change. Okay, those can be added on top of each other. So we get graded potentials there. Those are going to stimulate the next cell in line, which is called the bipolar cells. Okay, and then those bipolar cells also, when they get stimulated, they also produce graded potentials, not action potentials yet. And then finally, the last one, the ganglion cells. These are the ones that the axons are actually what make up the optic nerve. Okay, and that's actually where we're finally going to get an action potential, is those ganglion cells. And I had a question come through on the chat. Let me look at that. Would it ever be advantageous for light scattering to not be inhibited? Maybe say that a little bit different way, Bella. Is you're saying, would it be advantageous to not stop the light scattering? I think is what you're asking. Not so much. Yeah, I mean, in some ways, and that is why nocturnal animals don't have this, is because they want to let in as much light as possible, because they spend most of their time in the dark. And so they don't want that light to be soaked up by other parts of the eyeball. They want it to all get to the retina. And so in that way, in very dark situations, it is better to not have as much extra pigment in the eye. And so, yeah, so in dark situations, we don't want to absorb as much of light by the structures that aren't the photoreceptors. So I think that answers your question. Okay, so let's take a look at this one here. We can just walk through this. So the retina cells. So this is kind of zooming in on the retina, and we can start back here with these cells, the photoreceptor cells. So those would be rods, whereas B would be the cones. Okay, and then C. So again, we're getting graded potentials on those. Graded potential is going to activate these next ones called the bipolar cells. We get another graded potential on these bipolar cells. Those are going to then activate the ganglion cells, which then you can see their axons are just what make up the optic nerve. So they're all going to come together and then exit out of the eyeball. Okay, so that E is the optic nerve. Okay, so which ones actually send action potentials? Those would be D, the ganglion cells. Okay, and I'd like for you to think about this one for a second, that question there in the bottom. I have two arrows down there, one labeled as 1, the other one labeled as 2. I'd like you to send in on the chat in about 30 seconds the answer to the first question: which arrow is the direction of the light as it enters the retina? Would that be 1 or 2? Think about that for a second. Put it in and get ready to hit enter. And go ahead and hit enter there. Yeah, so mostly saying 1. We got a couple people saying 2. So it's actually 2. 2 is the direction of the light. Yeah, so only a few people said 2. Most people said 1. It's kind of a weird situation because the light has to pass over the ganglion cells, has to pass over the optic nerve, has to pass over the bipolar cells, and then it hits the photoreceptor cells. And then the signal goes back the other direction, right? So the signal is going back this direction, but the light had to pass over those other cells first. So it's just kind of an interesting setup. And that's why I have that question there, just to kind of bring it up, that's the way that those two things kind of travel. Part of the reason for that is because this is that pigmented layer back here, and the photoreceptor cells are the ones that need the pigment. So we need to be able to use the pigment, we need to be able to replenish the pigment in these photoreceptor cells. So they need to be right by the source of that. It's also because we need these cells, these photoreceptor cells, to be on a flat surface, right? If they were on top of these ganglion and bipolar cells, then it wouldn't be a smooth surface that the retina finds itself on. And so that's part of the reason for that. Okay, any questions on that? Okay. All right, so a little bit more into the retina. The retina has three primary different regions, and those are shown here. So again, this is the optic nerve, so it's the back of the eye. The part, there's a part here called the fovea centralis, okay? I'll just open up all of them. The periphery part is out here, okay? The periphery is where we find mostly rods, okay? So this is what gives us our peripheral vision, okay? So when we're looking forward at something, I'm looking at my finger right now, I can see that very clearly, but I can also see the things over here on both sides, right? And so the things I'm seeing on my sides, I'm seeing because of the light that's hitting the periphery of my retina, okay? But the thing I'm looking at right in the center, that light is going directly to this thing right here called the fovea centralis, okay? And the fovea centralis is where all of the cones in the eye are, right? So those 5 or 6 million cones, however many I had on that slide, those are all just densely packed in that one spot, okay? So the cones are really what give us our clear color vision, whereas the rods are better for peripheral vision and less clear, less acute vision. Okay, and then we have what's called the blind spot, or also known as the optic disc. That's just right here, because when light hits that spot where the optic nerve exits, we don't actually see that because that's just where all the axons have to exit, so there's no photoreceptor cells there. Okay. All right. So that's the retina. Let's talk a little bit about the different segments of the eyeball. The lens is really the dividing line between the anterior segment of the eye and the posterior segment. Okay, so the lens is very elastic, protein-packed structure, and when we stretch it out, it will bounce back to its original shape. That's the elasticity part of it. Okay, and so we can change the shape of this thing to focus the light. We'll talk more about how that works next time. But the light's already being bent a little bit by the cornea, and then it goes through the lens, and the lens kind of does the fine tuning of that light to make it focus perfectly on the retina where we want it to. And like I said, it also divides the anterior and posterior segments. Okay, this is just an image of an eyeball that was ruptured. Okay, I put that in there to remind me to tell a story of when I was working as an athletic trainer for the baseball team here at Point Loma. They were just having practice, and I was working in the clinic down there, and you know, they put those like that fence up like around the batters when they're doing batting practice, so it makes sure foul balls don't go towards the dugout and things like that. And one of the coaches was standing right around third base, but he was just far enough in the field of where that net doesn't protect. He was talking to somebody else, and the guy hit a line drive and just hit him directly in the eyeball. And he had glasses on, and this is pretty much what happened to his eyeball. He had a globe rupture. When that ball, something hits your eyeball that hard, it can literally just burst out the back of your the sclera in the back just burst open. And that's what happened. And when that happens, the lens falls backwards, right? And the lens is supposed to be way more anterior, and that lens falls backwards. The retina falls off of the back of the eye because you lose pressure. And so it's a devastating injury to the eye. But in his case, he was able to, this is a coach that's no longer here, it was a long time ago, but he went through like 7 different surgeries to kind of put his eye back together, and he was able to still see from his eye, but it's pretty gnarly when you have a globe rupture like that. That's not his eye there, but that was just one I found. But yeah, so yeah. So that's a side note, but it's always a fun story. Okay, so when we kind of already talked about the anterior part of the eye. That's the part that has that aqueous humor in it. But then the part behind the lens, the posterior, that's what has the vitreous humor. So it's much more jelly-like, but it's also very clear and transparent, so the light can pass right through it. Okay, we really only form our vitreous humor once during development, and we can kind of add to it and turn it over, but it takes a long time to build back up vitreous humor when you have an injury like this. And so it becomes challenging to gain that pressure back in the eye if you've lost some of the vitreous humor, because it really maintains the shape of our eye, holds our lens in place, as well as holds our retina in place just by creating some pressure in the posterior part of the eye. Okay. So then the anterior segment is on the other side of the lens. All right, so much smaller. We're just talking about this part up here. Okay. Again, it's full of aqueous humor, which is much more fluid-like. Okay. And it's constantly being formed and drained. And so it's made much quicker than vitreous humor, and it's constantly being replenished, and is secreted by those ciliary processes. And then the sclera venous sinus is a little track it takes to exit out of your eye. And in some cases that can get clogged. And then what can happen is the aqueous humor keeps getting produced, but if it's not able to exit the eyeball, then you get pressure in the eye. And that's really what this looks like here. And that's exactly what glaucoma is that it's the buildup of pressure in the anterior chamber of the eye, and it starts pushing the lens backwards, right? So it starts smashing the lens backwards, which then smashes the vitreous humor up against the retina, and you can have damage to the retina. And this is why people with diabetes are at much higher risk of blindness, is because the higher blood sugar you have, you end up clogging up the little tubular system that drains the aqueous humor. And so it gets blocked and you can't release the aqueous humor, and then it pushes that lens backwards. Yeah, so that's pretty much it. Any questions about any of that? All right, so let's talk a little bit about the muscles that move the eye. Okay, so these are known as extrinsic muscles. The intrinsic muscles are the ones we just talked about, the ones that control the shape of the pupil, or the size of the pupil and the shape of the lens. Then we have extrinsic muscles that actually move the eyeball, right? Move it up and down, move it all over. And we talked about these some last semester, and so this should be a review. We have the 4 recti muscles. All right, we've got the superior rectus, which moves our eye up. We got the inferior, which moves our eye down. We got the medial rectus, which moves our eyes medially. Lateral rectus, which moves our eyes laterally or out. Okay, cranial nerve 3 or oculomotor for those 3, and then we have cranial nerve 6 or abducens for lateral rectus. Remember, abducens literally means like abduction, so it causes abduction of your eye. It causes your eye to move outwards. That's the only thing that nerve does. It doesn't do anything else. Just controls one muscle. Okay, and then we got those 2 obliques, kind of these curved muscles. Okay, so the superior oblique pulls the front of the eye downward and lateral. Okay, best way to look at it is from the little image in your book right there, down and lateral. Okay, and then that inferior oblique does not exactly the opposite, but it pulls the front of the eye upwards and lateral. Okay, so it kind of makes you look up and out, whereas the superior oblique is down and out. Okay, you can see that again, one of those is controlled by oculomotor. So out of the 6 extrinsic eye muscles, 4 of them are controlled by the oculomotor nerve, 1 by the abducens, and 1 by the trochlear nerve. Okay. All right. Anybody? Yeah, it looks like a couple of you are still writing. I'll give you a second to finish writing that down. Any questions on any of these so far? Okay. Good to scroll through and see all your faces. Everybody paying attention. I like it. It'll be a lot more fun when I don't have to scroll through. I can just look at you. All right. Okay, so let's move on here. And yeah, why don't you take a second and see if you can label these guys. I should have had you doing that while I was waiting for everybody. Go ahead and see if you can label these muscles. And then in about a minute or so, I'll have you send in that letter of the one that moves the eye up and out. Okay, so think about that one for a second. Label those. And you got multiple views here, right? This one's a lateral view of the eye, but it has the lateral side of the skull taken off so you can see it. Right? This one's just an anterior view, and then this one is a superior view. All right, so whenever you're Ready there? Let's go ahead and have you send in that letter of the answer to that question down there at the bottom. Okay, so you're gonna go ahead and hit send or hit enter on that with the answer to this question here. So which letter is the muscle that moves the eye up and out? Go ahead. And then the chat. There we go. Okay, good. So everybody's pretty much saying A, and that one is called the superior oblique. Okay, let's take a look at these here. So we can see A here. The superior oblique has a super interesting path that it takes. You can see it comes up this way, goes through a little pulley, a little tendon that's a pulley, and then it takes a total change in direction and catches the eye that way. Right? That little pulley is called the trochlea, and that's why the trochlear nerve is called that. And so yeah, so that's the superior oblique. Okay, B would be the one that's straight on top, so that's the superior rectus. C would be the one lateral, so it's our lateral rectus. D is right on the bottom, so that's our inferior rectus. E is going to be that other oblique, which is the inferior oblique. And then lastly, F is on the medial side, so that's our medial rectus. Okay, so you definitely need to know the names, the locations, what the actions they do, and then yeah, I think that's it. I was trying to look for another one there. Names, locations, action. Oh yeah, and then the nerve that controls them. Yes, that was the other one I was looking for. Okay, any questions on those? All right, I'll give you a second to keep writing that down. Yeah, I see your question there, Kat. I'm not sure which one C was. Let's see what was C. C was the lateral rectus. Okay. All right. All right. So yeah, the next slide. Well, give me a thumbs up if you're done. Give me a thumbs down if you need some more time writing that down. Anybody need more time? We're good. Okay. All right. Okay, so this is just kind of a cool image that shows some of those muscles with a clear eyeball, so you can see how all of those interact with the eye. But yeah, nothing more than just another way to look at it there. Okay, and so what I'd like you to do for a second is walk through this kind of slide for a second. Just try to fill in as much of this as you can on your own. So which muscles would we use if we're tracking an object upwards, which means there's an object in front of us and it's going upwards? Which muscles would we do for that? And then add in the nerve as well, okay? Same thing if there's an object up here, we start tracking it downwards, which muscle, which nerve would be used, okay? Do that with each of those. I'll give you about a minute and a half or a couple minutes to do that, and then I'll come back and fill it in with you, okay? So go ahead and fill in as much of that as you can here for a sec. Okay, so let's walk through this here just for a sec. And yeah, we don't need to try to incorporate the obliques in this. It gets too complicated for this class. And let me see, let's see. Oh, did I mess that one up, Emma? Let me take a look at that. Oh yeah, thank you, Emma. I said that one backwards. Yeah, the answer to this question should have been inferior oblique, right? Up and out is, oh no, that's right. No, so upwards and out is inferior oblique. And I had said superior oblique for that. Give me a thumbs up if you caught that and you just didn't want to say anything. I know Emma did, thank you. Yeah, I just said it backwards. Thank you. So that should be inferior oblique. And so then that one was not A, but it was E, right? Yeah, everybody tracking with me? Sorry for the confusion. Any questions on that? Okay. All right. Okay, so let's walk through this. So when we track an object upwards, we're using our superior rectus muscles, and that's going to be cranial nerve 3. Okay, when we track an object downwards, we're going to be using our inferior rectus muscles, also going to be cranial nerve 3. Okay, we track an object to the right. Now we have to differentiate with which eye we're talking about. Okay, we talk about the right eye. We're tracking an object to the right. That means our right eye has to move lateral, right? So it's going to be our lateral rectus, which is going to be cranial nerve abducens. Which one was that, 6 or 4? I don't remember. Let's go back for a second. I just don't want to give you the wrong information again. Yes, that one is 6. Thank you. So cranial nerve 6. Okay, and then our left eye is going to be the medial rectus, which is cranial nerve 3. Okay, we track an object the other direction to the left. So then in our right eye now is going to go medial. So it's going to be the medial rectus, which is cranial nerve 3. Okay, left eye is going lateral. So we're going to go lateral rectus, which is cranial nerve 6 abducens. Okay, now if an object's moving towards us, both of our eyes move medially, right? So we're going to have the medial rectus on both. Again, that's cranial nerve 3. Okay, objects moving away from us, you might not think about it all the time, but both lateral rectus muscles are working when we do that because our eyes need to straighten out. Okay, so lateral rectus, again, cranial nerve 6. Okay, all right, any questions on those? Okay. All right, give me a thumbs down if you need a little bit more time here. Okay, that's fine. Take a sec. Yeah, so a couple questions on there. Yeah, so one question, the function of the anterior segment that contains aqueous humor. Yeah, the function of the aqueous humor is to provide nutrients to the lens in the anterior part of the eye. So it's kind of, think of it like our synovial fluid in our joints, how we put nutrients into that and we can get the nutrients out. It's also to maintain some pressure in the anterior chamber of the eye. And then there's another question there. 2 questions. One, do we need to know the specific cranial nerves or just the numbers? You need to know both the names, numbers, functions. You need to know all of that. And then another one, can you explain the last 2, please? Again, sure. So when an object is moving towards you, and you can do this yourself right now, take your finger and just bring it right towards you and stay focused on your finger. your eyeballs move like this. They both move medially, right? In order to stay focused on that finger, they both have to move medially. So both the medial rectus muscles, or the medial rectus muscles in both eyes, are activated to do that. Okay. And then if you start with something close and you're trying to focus on it, both your eyes are already medially moved. And so that when you move them out, when you move the finger out, your eyeballs do the opposite. They start moving laterally a little bit until a certain point, and then they're kind of just straight, but they move laterally first. So that's why we would use the lateral rectus muscles in both eyeballs first. Okay, any other questions there? You're welcome. Okay, so let's talk a little bit about how the eyeballs move. There's kind of a couple of different things going on here. This is a very simplistic way to look at it, what we just did there on that last slide. It's much more complex than that. All the muscles are working together to produce circular and diagonal type movements all the time, right? But they're very, very small motor units in these little muscles, right? These little skeletal muscles, and they have little motor units with just a few muscle cells in each. So we can do very fine, very precise little movements to control it. So one term that's important to know is this thing called saccadic movement. Oops, there we go, saccadic movement. And what this is this kind of rapid jumping from one focal point to the next. Okay, so the way this works is if you are, so for example, one way to think about it is picture yourself driving in a car, driving through the country. If you've been in some farm country, then you know what I'm talking about when you're driving through and you're just staring off to the side, and there's the rows of trees or rows of crops, right? Your eyeballs will tend to focus on one row so you can see down it, and then your eye moves with that until it can't see it anymore, and then it jumps back to the next one, right? And then it goes down until it can't see it anymore, and it jumps back to the next one, right? That's the saccadic movement, is rapid jumping from one focal point to the next, because there's no point in your eyeballs just moving quickly, because they can't focus on something when they're just moving like that. So instead, they find a focal point, stick with it until they can't stick with it anymore, and then they jump back, find another focal point, stick with it, and that's really what that saccadic movement is. Okay. Now it's a little bit different when we're tracking something. When there's an object actually moving in front of us. There's kind of two ways to look at this, and I'll explain this one a little bit more when we talk about the ears. Because the ears are involved in this. But when we're tracking an object that's moving, our eyeballs are able to stick with it if it's moving too, right? As long as it stays in our field of vision. But in the previous example, it moves out of our field of vision. That's why we have to jump and catch another thing that we can focus on. But we can track an object that's moving, and we can just stay focused on that same object the whole time. Yeah, so that's kind of that. And then we do have some abnormal movements. Strabismus is just kind of the medical term for cross-eyed. And then diplopia is the medical term for double vision. Okay, and that's really what happens when you have strabismus, because you have each eye seeing different things, right? And that's really what double vision is one eye sees one thing, the other eye sees another thing, and your brain's trying to deal with both of those images. And so you see two things, right? And that's really what double vision is. And that's what happens with strabismus, because your eyes aren't focused on the same spot, right? One eye might be focused here, the other one's focused there. So you're getting one vision from this eye, one vision from this eye. And so you see two things. If it happens early enough in development, then your brain can actually totally compensate for that, merge the two images together so you only see one. But if it just happens temporarily, then you will see two images for a while. So that's it, guys. All right, so have a great rest of your day. We'll see you on Friday. We'll move into the function of the eyes, more of the physiology. And yeah, you have all the material you need now for the quiz, so you can hop into that whenever you're ready. But again, study hard for that. Make sure you use Canvas as your browser, or sorry, use Chrome as your browser. Don't use any other type of browser for that. And you have an 18-minute time limit, so make sure you study. All right, we'll see you guys.
Thank you.
Thank you.
Thank you.
Thank you.
Thank you. Dr. Sorensen.